Mechanism of action of toxicants. Under this topic, today we will study mode of action of heavy metals. First of all, we will discuss generalized scheme for toxicity pathways. And secondly, we will discuss mechanism of action of heavy metals, which may be categorized as by affecting permeability of plasma membrane and of subcellular organelles, by stabilizing tissue constituents and cellular organelles, by inactivating enzymes and cell constituents, by binding with functional groups, by causing irreversible damage to nucleic acids after binding with them. The question is, what is heavy metal? A heavy metal has a high atomic weight with a specific gravity that exceeds the specific gravity of water by 5 or more times at 4 degrees Celsius. Major toxic metals may cause multiple effects, while some are less toxic. Essential metals also exhibited potential for toxicity. Toxic metals and metalloids include arsenic, beryllium, cadmium, chromium, mercury, lead, nickel, and many more. These metals have various sources in the environment, like agriculture through fertilizers and pesticides, metallic waste disposal, metalliferous mining, and some industrial process like smelting and electroplating, etc. Metals from these sources enter plants and animals' body, causing severe damage to them. To understand how a toxicant enters an organism and how it interacts with target molecules, consider that toxicants entered the animal body, their presence is unbearable by the body, then they interact with target molecules and cause alteration of biological environment, then cause cellular dysfunction and sometimes stop the body from repair process. So, it adopts 3 pathways while causing damage to living organisms. The most direct pathway, known as Path A, occurs when the chemical causes toxicity by its mere presence at critical sites in the body without interacting with the target molecule. For example, when agents precipitate in renal tubules and block urine formation. An example of more complex route, Path B, is the toxicity caused by the tetrodotoxin, fish poison. After ingestion, this poison reaches the Na+ channels of motor neurons, blockage them and inhibit the activity of motor neurons, and ultimately skeletal muscle paralysis. No repair mechanisms can prevent the onset of such toxicity. When the perturbations induced by the toxicant exceed the repair capacity, or when repair becomes malfunctional, toxicity occurs, which comprises of tissue necrosis, cancer, fibrosis are examples of chemically induced toxicities whose development follow this 4-step course. In animal bodies, reactive oxygen species are produced due to endogenous and exogenous sources. Antioxidants work against these reactive oxygen species to maintain balance between their production and repair, failing which leads to deteriorating effects on DNA, protein, and lipid molecules. Here is mechanism of action of heavy metals, which may be categorized as by affecting permeability of plasma membrane and of subcellular organelles, by stabilizing tissue constituents and cellular organelles, by inactivating enzymes and cell constituents, by binding with functional groups, by causing irreversible damage to nucleic acids after binding with them. Coming to first point, that is, metals cause toxicity by affecting permeability of plasma membrane and of subcellular organelles. As some metals, viz., Cu, Hg, etc., interact with phosphate fractions of plasma membrane, bind firmly with thioligands and exert conformational changes in the membrane. As such changes may either activate, exerting a series of cell responses resulting in cell damage. Lipid and protein molecules are the chief component of plasma membrane. Various metals like Cu, Ar, Mn, Ni, Co and Hg may bind with phospholipid fraction of plasma membrane and cause following effects. Hb and Pb increase ion permeability, Hg block glucose transfer into red blood cells. Coming to second point, that is, metals cause toxicity by stabilizing tissue constituents in cellular organelles. A number of metals bind with tissue components, stabilize them, and thus exert toxicity. For example, Cu and Hg bind with collagen protein Co. Ni, Cu and Zn form covalent linking with protein by binding with imidazoles nitrogen of histidine, and Sh groups of cysteine. Hg and Ag bind with Hb irreversibly and form HgHb and AgHb complexes, respectively, and then obstruct O2 carrying function of hemoglobin. 4. Hg also binds serum albumin and stabilizes it. Metals show affinity with NH2 group. These bind with NH2 groups of protein, causing irreversible change. The affinity of metals for NH2 group is in the following order: HgCuNi PbZn Co CdMn. Coming to second point, that is, Metals cause toxicity by inactivating enzymes and cell constituents by binding with functional groups. Metal ions bind with functional groups or ligands present in biologically active molecules and form coordinate covalent complexes with nitrogen ligands or other groups such as. Examples: lead Pb binds with SH2 and H2 inside hemoglobin and causes lysis of porphyrin. It also binds with PO4-3 ligands and produces deleterious effects. CD binds with SH group of glutamic dehydrogenase and consequently causes irreversible inactivation of the enzyme and inhibits alkaline phosphatase and ATPase activity. Arsenic binds with SH group of hemoglobin and causes hemolysis. Heavy metal ions, in particular DHG2+, Pb2+, and CD2+, act as effective enzyme inhibitors. They have affinity for sulfur-containing ligands, e.g., Ss3-NSH and methionine and cysteine amino acids, which are part of the enzyme structure. Metalloenzymes contain metals in their structures. Their action is inhibited when one metal ion of a metalloenzyme is replaced by another metal ion of similar size and charge. Thus, Zn++ in some metalloenzymes is substituted by Cd++ through competitive displacement, which leads to cadmium toxicity. Cadmium-inhibited enzymes include adenosine triphosphatase, alcohol dehydrogenase, amylase, carbonic anhydrase, glutamic, oxaloacetic transaminase. Alide-inhibited enzymes include acetylcholinesterase, carbonic anhydrase, cytochrome, oxidase, alkaline phosphatase, ATPase, key enzymes used in the synthesis of heme. Metals cause toxicity by causing irreversible damage to nucleic acids after binding with them many metals. For example, CD, Pb, and L stabilize RNA. Certain metals like Zn cleaves 5-phosphate bond in RNA, resulting in depolarization. Cu and Cd compete with Mg+2 of RNA polymerase enzyme system, causing damage to RNA synthesis.